Let us discuss about the factors influencing the GFR. There are three important factors. One is a filtration barrier. Then we have the forces which are acting across the filtration membrane. Finally, the amount of blood flowing to the kidneys, that is the renal blood flow. We will see one by one. Now, what is this filtration barrier? So, let us quickly revise the anatomy, functional anatomy of the kidneys and the glomerulus especially. See, the blood vessel which is coming into the glomerulus is via the afferent arteriole. This is the afferent arteriole. So, this is the structure of the glomerulus and the Bowman's capsule. Okay. So, we have got the afferent arteriole which gives the blood to the glomerular capillaries. The arteriole then loops here as glomerular capillaries. This is the tuft of capillaries. This tuft of capillaries sits in a cup-like space called as the Bowman space. Okay. And this Bowman space, this is said to be the urinary pole. Okay. So, this Bowman space, now it continues as a proximal convoluted tubule. The afferent arteriole is having an endothelium. It's an arteriole, so it should have an endothelium. And there are certain specialized cells called as juxtaglomerular cells. We will be seeing about the significance soon. And the tuft of capillaries emerge out of the glomerulus Bowman space as efferent arteriole, right? Now, this continues as a peritubular capillaries. This tuft of capillaries is what is important for our glomerular filtration. Okay, you can see some projections, dark celled projections. These are the cells. Okay, these cells are called as podocytes. Okay, these are called as podocytes. So, the same glomerulus, you can see here it's like a cut section of the glomerular capillaries. There in the previous picture we saw the loops of capillaries. Now, it is just a cross section. You can see the capillary. This is one capillary. This is one capillary. This is a cross section, right? You can see the green color cells which is present surrounding the capillaries. These are the podocytes. Okay. Podocytes, actually it wraps around the capillaries with the help of its processes, podocyte foot processes. Okay. Other than that, you can see the afferent arteriole here and the efferent arteriole, the specialized cells of the afferent arteriole, that is the juxtaglomerular cells, and you can also see a tubule, that is the distal convoluted tubule. When it comes and loops around the glomerulus, they form a specialized cell called as macula densa cell. This macula densa cell, this juxtaglomerular cell, and there is some mesangial cells here in the glomerulus. Altogether they form the juxtaglomerular apparatus. We will see the significance soon. Yes, this picture is in fact an enlarged picture of the glomerular capillaries and the associated cells. Okay. So, here you can see this is a capillary. Can you see the green color cell here? This is the green color cell. This is the podocyte. This podocyte is actually projecting, or it is wrapping around the capillary. All this green color finger like projections are nothing but the foot processes of the podocytes. Okay. And you can see an yellow color cell here. This is a mesangial cell, that is actually the capillary. They are present in between the capillaries. These are the mesangial cells, and that has also got some significance. We will discuss later. Right. This is still more an enlarged picture of the podocyte and the capillary. You can see this is one capillary wall. You can see here the capillary wall is having lot of pores. Okay. So, thus that represents the fenestrated endothelium of the capillary. Okay. Fenestrated means it is having pores. Okay, through which the substances get filtered. So, this is the fenestrated endothelium. On top of on the outer layer of the fenestrated endothelium, you have got a basal lamina. This is a layer, a basement membrane which is called a basal lamina, just lying outside the endothelium. Okay, and this is the podocyte. Okay, this is the podocyte cell. It continues like this. One cell is here. And this is a primary process of the podocyte. You can see the primary process of the podocyte. Each primary process now spreads its finger-like projections that goes and wraps around the basal lamina. These finger-like projections are called as foot processes. Okay. Now, this particular cut section will be enlarged in the next picture. See, this is the picture. You can see the secondary processes. These are the secondary processes. You can see the enlarged foot process which goes and touches, or it goes and merges with the basal lamina or the basement membrane here. Okay. You can see the endothelium here, endothelial cells. These are the endothelial cells. In between the endothelial cells, you have got the gaps. Okay. So, these are the fenestrations. These are actually forming the filtration slits or fenestration, and here is a capillary lumen where you have got the blood.